Okay, so we're going to start to take a look at the eye today and what parts of the eye you actually have to be able to understand. And remember, it's kind of all about the structure and what is the function of the structure, and of course the location. So if we take a look at our diagram of the eyeball here, I'm just going to circle in red the parts that we actually have to know, because there are a few extra things here that aren't actually in our program of studies. So we do need to know the sclera, which is the white part of your eye, which is a fibrous tissue that gives your eye shape and helps maintain structure. There is the choroid, which is the concentration of blood vessels in the eye. So you can see that here, all of that network of blood vessels there. There is the retina, which is at the back of the eye, and that contains what we call the photoreceptors. Those are the receptors called rods and cones that help receive light and convert it into an electrical impulse. Then we have the fovea centralis, or sometimes it's called the fovea, and that's where the concentration of photoreceptors is the highest, and therefore visual acuity is the highest as well. We have the optic nerve, that's fair game. That's the nerve, of course, that transmits the action potential from the eye to the thalamus of the brain for interpretation. We have this structure right in here, which is called the blind spot. And it's called the blind spot because it actually doesn't have any photoreceptors there. Okay, so that's where the name comes from. Over on the other side here, we have the cornea. The cornea is the clear outer part of the eye that is responsible for the initial point of refraction of the light as it comes into the eyeball. The iris is the colored part of the eye, which actually has sphincter-like muscles that can contract to control the size of the pupil. So the pupil, therefore, is the opening. It literally is a hole in the eyeball that allows the light to pass through to the next structure, which is the lens. And the lens is the second point of refraction of the light as it comes into the eye. So it's bent initially by the cornea, and then secondarily by the lens. And then the last thing that we have here that we need to know is the vitreous humor. And the vitreous humor is the jelly-like substance that is in the cavity of the eyeball. And what that allows us to do, or allows our eyes to do, is to maintain shape and turgidity and structure. So now we'll look at the different parts of the eye in just a little more detail here. So we can take a look at, in our graphic here, of the cornea. So the cornea, as we said, is that clear part on the front of the eye, so you can see it there. And then you can also see the cornea on the other side of the eye. You can see it, that it covers the eyeball here. And as we said, that is the initial point of refraction of the light as it comes into the eye. Okay, and so if you kind of look at the eyeball from a side view, then you can see that it actually has that clear lens in front of it called the cornea. The cornea, incidentally, is the only tissue in the human body that can be transplanted without fear of rejection from donor to recipient, because there's no immune system response to that particular tissue. So that's kind of interesting. Okay, so now we'll talk about the pupil. As we said, the pupil literally is a hole in the eye that can adjust in size. It's sometimes also called the aperture. It can adjust in size to allow maximum amount of light in or out. So obviously your pupils are going to dilate, as shown here, when it's darker and there's not as much light available. Okay, so the pupils will dilate to allow more light in, and then of course pupils will constrict in bright light to restrict the amount of light that actually is allowed into the eye. And when we talk about the control of that pupil, or control of the size of that aperture, then we have to talk about the iris. So the iris is that colored part of your eye, and if you look closely at it, you'll notice that it almost looks like it has these lines radiating out from the center of the pupil, and those are actually ciliary muscles. It's almost like a sphincter that well, it is a sphincter muscle, and what it does is as the light increases or decreases, it's actually a reflex response that those muscles will constrict or relax in order to encourage the pupil to get smaller or larger, depending on the amount of light that is available. Okay, so it actually is that colored part of your eye that controls the size of that aperture or the size of that hole, that pupil. So the next part that we talk about is the sclera, and the sclera is a fibrous tissue. That means it's tough and it's dense, and it's that white tissue that covers the eyeball, so we can see that over here. The sclera has blood vessels in it, and obviously because it needs oxygen and nutrients, etc. And it actually covers all parts of the eye except the cornea, okay? So it's for protection, it's for support, and it's also for muscle attachment. The lens is, as we mentioned in the first slide, our secondary point of refraction of light. So as light passes into the eye, it's first going to be bent by the cornea, and then it's going to be bent further by that pupil as the light passes in. It's what we call a converging lens, and that means that it allows us to see objects both near and far. So the size of the lens will actually change. It'll flatten out or it'll get larger in order to accommodate when we talk about near and far distances, okay? So it actually has the ability, because of these muscles that are attached to it, these little ligaments and muscles attached to it, it actually has the ability to flatten or to kind of round out more like a Skittle, a little candy, so that we can adjust our vision for when we're looking at things far away versus when we're looking at things closer to us. The next part that we'll talk about is the retina. And one thing to remember about the retina is you can see here in this image of the retina. So sometimes if you go to the eye doctor, they always want to check the health of your retina. They're always looking for retinal disease. They're looking for a detached retina, which means that the retina literally starts to pull off the back of the eye, and then that optic nerve detaches, and that can result in blindness. And one of the hallmark characteristics of an onset of a detached retina is that you'll have momentarily have like flashes, or momentarily be blind, like just for a snapshot, super quick, or have like crazy flashes that happen super quick and then go away. Those are all indicators of a potentially detached retina. Now, we don't see detached retinas in young people very often unless there's an accident. So if someone takes a blow to the head, and it can actually result in a detached retina just because of the impact. But typically speaking, what happens as people get older is the vitreous humor, which remember is that jelly-like liquid that is inside of the eyeball that helps it maintain its shape and its structure, that actually starts to break down as people age, and therefore that eyeball becomes less turgid, less like firm, and then as things kind of degrade in there, that can actually cause the pressure to reduce and then that retina to actually detach. And if that happens, then the optic nerve is no longer getting information from the rods and the cones, from those photoreceptors that are inside the eye, and therefore communication is not going to the brain. Because remember, your brain is the one, your brain is what actually interprets what your eyeballs are transmitting. Your eye's job is just to take the light and create an image at the back of the eyeball, and then the rods and the cones convert that into an electrical impulse or an action potential, and then it travels along the optic nerve, but it actually doesn't get interpreted until it reaches your thalamus, and then your brain actually interprets what you're looking at. Now, obviously that happens really fast, but it is your brain that is making that interpretation, not the eyeball itself. The eyeball is just taking information in from the environment and transmitting it to the appropriate part of the brain. So you can see that the retina has a vast network of blood vessels in it, okay, so blood vessel rich for oxygen and nutrients. And then this is where we actually have those photoreceptor cells. There's two different kinds of photoreceptor cells, two different kinds. We have rods and we have cones. Rods are for night vision and cones are for color vision, and we'll talk about those a little bit later. Now, one thing that's super important about the retina is that the retina and the rods and the cones are responsible for converting light energy into an action potential. Because think about that for a minute. The optic nerve is a nerve, okay? So the only language a nerve speaks is an action potential. Neurons do not speak any other language. They don't speak light, they don't speak sound, they don't speak smell. The only thing they speak is action potential, that electrical impulse. And as a result, the rods and cones literally convert the light that's being brought into the back of the eye, and they convert that in a series of reactions to an action potential. And then once that has been done, then we have, of course, that action potential being transmitted along that optic nerve to the appropriate part of the brain. And then the brain, of course, because it's made of neurons, is going to interpret that response. So the next thing we'll talk about, just continuing with the retina, is just a little more detail in terms of the rods and the cones. So as we said, the retina is the very back of the eye. It contains those photoreceptor cells, and remember what that means. Photo always means light, and then of course receptor means that you're receiving something, right? So we have rods for night vision, we have cones for color vision. So if somebody is color blind, depending on the type of color blindness we're talking about, people that are fully color blind, meaning that they only see grays and whites and black shades of those variations, we would suspect that there is something wrong with their cones, okay, because they're not able to perceive light. So photoreceptors respond to different wavelengths of light, and again, they change the wavelengths of those light to an action potential so that the optic nerve can transmit it to the occipital lobe for interpretation. So this should look familiar. This is our electromagnetic spectrum, and remember, our part of the visible light spectrum is super, super small. It's just this little section right here, and then of course, if we pass white light through through a prism, then we get, of course, all those colors of the rainbow spreading out. And that's really what your eye can actually detect, is any type of light in that 400 to 700 meter or nanometer rather range, which is a very small part of the electromagnetic spectrum. So when we look at the difference between rods and cones, we again kind of just have to know, generally speaking, what the role of the rods and the cones are, okay, individually. And probably the best way to do that is to just look at our little chart here. So we have in our first column, we have the different categories here. So we have shape, prevalence, incidence by species, etc., etc. So which ones are really important for us? Well, shape is kind of important. The rods are almost like cylinders, whereas cones are kind of tapered at one end. So you can think of like how an ice cream cone starts out kind of in a round shape, but then it tapers to one end. The next bullet is very, very important. So this one here, prevalence in the human retina. So one thing I want you to notice is rods, the number of rods that we have is much, much greater than the number of cones. So the number of rods that we have for operating in dim light is much, much greater than the ones that operate in bright light to identify color. And if you think about it for a minute, when you are outside at nighttime and it's like really, really dark and there's not a lot of light, okay, there's no like lights on outside or fire or whatever, it's really, really dark, you can kind of see shapes, but everything kind of looks black and white or gray, right? You can't really see vivid color at nighttime, and that's because the cones are not stimulated unless there is bright light. So when you're walking around and it's dark, or when you wake up at night in your room and your eyes adjust and then you can kind of see the outline of the stuff in your room, that's the rods that are actually working there to help you be able to see in dim light. But you should notice that you can't actually see color when it's really dark. Even if you can see the shapes of things or the outlines of things, you can't see color. So that's very, very important. And of course, the concentration of rods in the retina much greater than the concentration of cones. In terms of species, it kind of stands to reason. This isn't something in our program of studies, but it's interesting. In terms of species, obviously, nocturnal species are going to have a very high concentration of rods, and diurnal species, or species that are active during the day, they will have cones. So there are some nocturnal species that don't have any cones at all because they've adapted to be active at nighttime, not during the day, and therefore the prevalence of cones is not useful to them, and over time have been eliminated altogether. So the area of the retina, in terms of where those rods and cones are, the rods are kind of on the periphery, so the sides, and then the cones are more towards the fovea. And remember, that's where we have the highest amount of visual acuity. So the cones are more concentrated around the fovea centralis of the retina, whereas the rods are more along the periphery or the sides, okay? The next bullet is important, contribution to color vision. Remember, rods have no direct contribution, and obviously cones are critical. Excuse me. So as we said, individuals that are colorblind, especially the ones that are completely colorblind, they do not have cones that are functioning properly in their retina. Response to dim light is very strong for rods and very weak for cones, and contribution to perception in detail. Okay, so think about that. When we talked about like it being dark in your room and you can kind of see the outline of things, like you might recognize that, oh yeah, that's my lamp, and oh yeah, that's my bookshelf, but you can't really see a lot of detail, right? And that's because rods do not contribute to detailed perception, okay? Whereas in brighter light, cones give you much more detailed perception, not only in terms of color, but also in terms of resolution, or how clearly you can actually see something. So continuing along with the optic nerve, we've talked about the optic nerve a little bit already, and when we do our eyeball dissection, we'll be able to see the optic nerve very clearly. So here's a mammalian eyeball that has been removed from some type of mammalian species. You'll find that this actually does kind of look like a human eye, but in any case, coming out the back of the eyeball, you'll see the optic nerve, okay? Now this is a nerve. Remember what we talked about when we talked about neurons. Neurons only speak in action potential, okay? They do not speak in any other language. And so remember, the whole point of those rods and those cones is they facilitate the conversion of light energy into an action potential, so that the action potential can be transmitted to the thalamus, eventually to the occipital lobe for interpretation, okay? And you can see where that is in your brain here. So it's kind of freaky looking. So this is the front of your brain, right? And then you can see that here's the visual cortex. So that's like where the occipital lobe is, right? And you can see that we have this big optic nerve traveling out the back of that eyeball, yeah? And then we have that information being transmitted to that visual cortex in the form of an action potential for interpretation. So it's not until that action potential gets to the visual cortex here and that occipital lobe that it is actually interpreted in terms of what you're seeing. Now obviously that happens very, very quickly. Remember, you know, neurons can transmit 400 kilometers, 500 kilometers an hour, or sorry, so it's super, or per second rather, so it's super, super fast, right? So that all happens very, very quickly. There's no like lull in sort of time in terms of that information getting to your brain and your brain interpreting that, okay? Now, the other thing that's important about the optic nerve as well is where the optic nerve attaches to the back of the retina, okay? So we can look at that right here. So where the optic nerve attaches to the back of the retina, that's actually the blind spot, because if you think about it, you don't have a bunch of rods and cones there because that optic nerve is actually attached to the retina at the back there. So that is what we call our blind spot for that reason. The other thing is remember when the image is, excuse me, created at the back of your eye, excuse me, when it's created at the back of your eye, we actually have the image inverted, okay, it's upside down, and then again, not until it gets to that visual cortex is it actually flipped and put right side up. So we talked about the blind spot already. So again, you can see in this image here where the optic nerve attaches to the back of the retina. There's no rods or cones there, and therefore we call that the blind spot because there's no receiving of light or converting that light